Hello students, today we will start about new topic, that is linkage. Linkage is the term that we apply for genes or alleles that are present on same chromosome. So if you see diagrammatically, if this is the chromosome and here are some types of genes like they are responsible for one character and another type of genes and these are responsible for some other character like color or height or anything, then we call these two genes or allele forms as linked genes or linkage group. So linkage can be defined as the physical association of genes on a chromosome. Always I am using on a chromosome, not between two different chromosomes. It is not between two different chromosomes. Again, we can also use some other sentence to explain linkage as linkage is the tendency of genes to exist on same chromosome and those genes remain together during inheritance. What is inheritance? The process of transferring of characters from parents to offsprings, means from one generation to other generation without any change or without separation, means they are not getting separated. Those linked genes are not getting separated during the process of inheritance. Next, the people who studied on linkage for the first time are BATESON and PUNNTTE. So, we give a term like linkage is first discovered by BATESON and PUNNTTE. They have done work on sweet pea. Now, sweet pea scientific or botanical name is Lathyrus odoratus. Lathyrus odoratus. Now, this is different from another type of example that was studied by Mendel. Mendel has done his work on pea plant. We give a scientific name for this as Pisum sativum. This is different from sweet pea. Its scientific name is Pisum sativum, but its botanical or scientific name is Lathyrus odoratus. Then, Lathyrus odoratus is also called as sweet pea. Then, what is another name for Pisum sativum? It is also called as garden pea. So, Mendel has done work on this one, but BATESON and PUNNTTE has done work on sweet pea, that is Lathyrus odoratus, in the year 1906. Linkage is experimentally proved by Morgan, T.H. Morgan. T.H. Morgan. So, we give a term for Morgan as father of experimental genetics. In Drosophila we also call as fruit fly. Now, all genes are located on same chromosomes are said to be linked genes, which I told you initially. This linkage, it is an exception of law of independent assortment. What is independent assortment? It is the third law given by John Gregor Mendel. In this, we read few examples that if a pair of dominant alleles which are responsible for two different characters and are responsible for dihybrid cross are present in an inheritance, then each dominant allele or each dominant gene will be expressed without concern of existence of other dominant gene beside it. This law of independent assortment, this is not applicable for the studies done by BATESON and PUNNTTE because here we read about the alleles that are present on different chromosomes, not on same chromosomes. So we say this is an exception. Why exception? Because law of independent assortment is applicable for genes on different chromosomes. According to law of dominance and the first law given by Mendel and law of independent assortment given by John Gregor Mendel, these two are not universally applicable. BATESON and PUNNTTE selected two characters of sweet pea that is different from Mendel's dihybrid ratio, or that is different from dihybrid cross. Now, they explain the total work done by them with the heading coupling and repulsion hypothesis. So, they explained linkage as coupling and a repulsion hypothesis. In year 1906 itself. So for their experiment, they have taken nearly 2132. This is just an approximate number. I think it is not the exact number, but it can be taken as an approximate number to explain the work done by Bateson and Punnette. They have taken 2132 number of sweet pea plants. For their experiment. So, expectation is, expected ratio we will write 9:3:3:1. That we know according to dihybrid cross that we have done just before when we are reading about law of independent assortment. Next, but the ratio that they have obtained is quite different. That is, they have seen a ratio of 14:1:1:3. Means here purely dominant alleles are expressed, here recombinants are expressed, and here recessive one are highly expressed here. In 9:3:3:1 ratio also, you will see here mostly dominant, both pairs or single one is getting expressed. Here, this 3 and this 3 represent the recombinants, and this one represents the last recessive expression of the alleles in dihybrid cross. Now, they did what characters? We already have seen they have selected two characters just before. So, what are the two characters? Two characters selected by Bateson and Punnette are, one is blue flower with long pollen. So this is one character. According to the morphology of pollen grain, this is another character. So we also know there will be some contrasting features. So the contrasting traits are, if one blue color flower is considered as dominant, then red color flower will be considered as recessive one with round pollen. So these are the two characters selected by Bateson and Punnette. So in F1 generation, purely they also got blue color flowers with long pollen in all the offsprings. But when they have self-crossed, again the F1 generation, to observe the F2 generation, then they got some different ratio that is not seen in case of normal Mendel's dihybrid cross. We will see those types of the variations also, how the 14:1:1:3 type of ratio was obtained from a total collection of 2132 number of plants. So the phenotypic ratio in F2 generation, we have seen that the ratio is not 9:3:3:1. So the expected will be ratio is 9:3:3:1. Then what about the observed one? Observed ratio 14:1:1:3. Now, what are 14 number and what is the phenotypic expression of those 14 number of flowers? Blue flowers with a long pollen. They are said as 14, not 14 in a number, that is a ratio. This is one numerical value. This distribution, after this I will write here. Next, blue flower plus short pollen 1 of the total ratio. Next, red flower again, this is also 1 according to the ratio. Next, red flower actually this last one, purely recessive of phenotypes, we should get very less number comparing to the other ratio numericals. But here we got a little bit more comparing to the recombinants. Here, expected I have written. So we will write some numerical distribution of total number of 2132 number of plants. So expected out of 2132 number of plants is blue color flower with a long pollen. Let's give some symbols before writing there. So this is BL with small l, small b, capital L, and small s. According to 9:3:3:1 ratio, this blue color flower with long pollen, their number out of 2132 should be 1199 in number. This is equal to the ratio 9. Next, blue color with a short pollen, this number should be 400, which is equal to 3. And red color long pollen, this number also should be 400 according to the ratio 3. And purely recessive one, its number should be 133, so that we'll get the ratio last as 1. But the obtained number is different. Observed ratio is blue color long pollen is 1528, which is equal to 14 of the ratio numbers. Blue color round pollen is 106 in number. Red color long pollen is 117 number. Next, purely recessive one is 381 number, which is equal to 3. So this is the expected number and this is the observed number. So in Mendel's dihybrid ratio, law of independent assortment was involved and both dominant alleles got expressed with concern of the other dominant allele. So they expected this ratio. But here, when they have observed this one, means more number comparing to the 9, here it is 14. So they thought that there is more linkage between the parents, so that only the dominant type of alleles are much expressed in their experiment by taking sweet pea or Lathyrus odoratus. That I am going to write here. So in Mendel's dihybrid cross, law of independent assortment was involved. So both dominant alleles expressed their characters. independently. So they got ratio as 9:3:3:1. But Punnette's Bateson and Punnette has observed instead of 9 and instead of 1119 number of dominantly expressing charactered plants, they have observed more, that is 1528 number. So they thought that here parental combinations are more or highly expressed. This is all because of the linkage between dominant genes. By obtaining the 14 number instead of 9 according to the ratios, according to Mendel's dihybrid, 9:3:3:1. But they have seen 14:1:1:3 number of ratios. So whatever the number that has been increased instead of 9, that is 14, that 14 number of dominant expressed genes, that gave an opinion that there is more linkage between the parents. But they could not prove the reason. They also conducted same type of experiment by taking test cross. So what is meant by test cross? Any of the parent or F1 generation or F2 generation or any unknown type of genotype containing allele forms are taken for experiment. They are crossed with pure recessive allele forms of the genes. For suppose, if we have taken a dihybrid cross or any monohybrid cross like this, for suppose this is dihybrid and this is monohybrid, just think for a while. It has to be crossed with purely recessive allele forms or it has to be crossed with purely recessive allele forms. So such type of cross is called as test cross. Test cross can be defined as the cross that is done with an unknown or any known type of genotypes purely with recessive allele forms. So for this test cross, they have taken 2 flowers. They have done 2 types of collections for their test cross. One is they have taken 2 flowers from the same plant and they took care that the chromosomes which are involving in the test, means homologous chromosomes that are formed from the a type of zygote that is fused by 2 identical gametes. Here they took care that only one chromosome of the homologous pair is having both dominant alleles and another second chromosome of the homologous pair that has purely recessive alleles. They took care that the genotypic distribution is like this by taking 2 flowers from the same plant. They also took another experimental flowers that are 2 separate flowers from 2 different parents. So how here the chromosomal or the allele distribution in the chromosomes is means, if these are the homologous chromosomes, then here is one dominant allele pair and in the second chromosome here is another dominant allele. 2 dominant alleles are not distributed on same chromosome. They took these 2 types of flowers for their experiments and they have done cross. We will see about the results that are obtained after doing this test cross between 2 different types of parents. Here we can consider the first experimental flowers as purely original flowers because naturally we see such types of flowers only. Original plant we can consider. The second experimental flowers that we are taking here according to the dominant allele distribution, we can consider these flowers as crossed flowers, means that are obtained in the F1 generation. Crossed flowers that are having dominant alleles on 2 different chromosomes. Let's see now the detailed explanation of 2 experiments done by Bateson and Punnette in the test cross. So first example is by taking 2 separate flowers of the same plant. So when we see the homologous distribution of alleles in chromosomes. So as parents, Bateson and Punnette have taken blue colored flowers which are considered to be dominant with long pollen. Now, which are crossed with red color flowers with round pollen. These both blue color flower with long pollen are dominant alleles or dominant characters or traits. These both red flowers and round pollen are considered to be recessive traits. And their genotypic distribution is considered as capital B, capital B, capital L, capital L. And here recessive one is considered as small b, small b, small l, small l. If we see how this type of allele distribution is there in the flowers that I have taken or that are taken from the same type of plant, both dominant alleles which are responsible for blue color and which are responsible for long pollen are present on the same chromosome. And the recessive one which are not expressed in the next generation are present on other second chromosome. If we consider these both as homologous chromosomes, the arrangement of alleles like this, as all dominant on one chromosome, all recessive on other chromosome, we call such type of allele arrangement as cis arrangement of alleles. Or alleles are arranged in cis pattern or cis form. If we see the F1 generation from these parents, we will get all genotypes as capital B and capital L means dominant allele for long pollen, dominant allele for blue color are present on same offspring, and we will get all offsprings of blue color with the long pollen. Then, as the experiment is nothing but test cross, it has to be crossed with the recessive allele forms. Then, what is happening in the F2 generation? In F2 generation, if we consider the sample size, suppose sample size taken by Mendel, and here the number of offsprings are 16. Here, according to Mendel, that he has taken here, sample size is approximately like this. And okay, offsprings are 16. Then the test cross ratio is 4:4:4:4. When we simplify this, the ratio is 1:1:1:1. But according to Bateson and Punnette experiment, sample size is 2132, that we are telling approximately, not the exact value. Here it is 24,000. Okay. Approximate value, not exact number, that is out of the sample. Here, more offsprings are of parental type. We cannot even distribute like this also. Parental type. We'll write that one. 931 number are of parental type. 133 are of blue color flower with round pollen. And again 133 number are of red color flower with long pollen. Again 931 are of purely red color flower with round pollen. If again we simplify this, the ratio is different, that is 7:1:1:7. Here, these both Middle 1 and 1 are considered to be recombinants. Recombinants. First 7, next 7 are considered to be parental type. Parental type. So here it is proved that parental type even after test cross are more in number comparing to the recombinant offsprings. This type of cross is given a term as coupling phase. Coupling phase, that is due to arrangement of alleles like the cis form or cis arrangement of the alleles. This is the first experiment done by Bateson and Morgan basing on the test cross. Clearly, I am telling you once again, basing on the 16 number of offsprings, according to Mendel, the ratio is 1:1:1:1. But Bateson and Punnett got different type of ratio, that is 7:1:1:7. The endings, first 7 and this one are representing the parental type. The middle 1 and 1 are representing the recombinant type. We will see the second type of experiment also. In case of second type of test cross experiment, the samples taken are from 2 separate pea plants. How the chromosomal distribution of alleles is present in such type? Here, the parents taken are blue flower with round pollen, that is crossed with red flower with long pollen. According to this, the allele distribution is blue color are indicated with capital B, round pollen are indicated with small l and small. Red color flower are indicated with small b, long pollen with capital L's. If you see the same type of allele distribution here, capital B can be in one chromosome of the homologous pair, and here another one, long pollen genotype or alleles are distributed or are present on other chromosome of the homologous pair. So here it will be like this and it will be like this. In the F1 generation, again the same type, capital B, capital L, small l. All offsprings are of blue colored flowers with long pollen. Even in first sample of coupling phase also we have seen the same type of result in F1 generation. Coming to F2 generation. To obtain the F2 generation, we have to cross this F1 generation with the purely recessive type of alleles. Then what is the result? If we consider, already we know the dihybrid cross according to Mendel, the number of offsprings are 16 in number. So, from 16 offsprings, according to Mendel, the ratio is 4:4:4:4. If we simplify this, we are getting ratio 1:1:1:1. This is expected ratio by Bateson and Punnett in the same type of cross. But out of the total samples collection that we see, but according to sample size taken by Bateson and Punnett, (133) according to the sample collection, is to 931 is to 931 is to again 133. The ratio is 1 is to 7 is to 7 is to 1. Now, here more number of recombinants. This 7 is to 7 is indicating recombinants. It means that the parental types are very less. Means the allele, there is more repulsion between the alleles which are present on separate chromosomes. So, this one and this one indicates parental type. As parental types are very less in ratio, this gives a proof that there is repulsion between the dominant alleles that are present on separate chromosomes. So we give a name for this type of experiment by taking two separate flowers from two different plants of sweet pea as repulsion phase. Repulsion phase. So this is the work done by Bateson and Punnette. Next we will see in the next part the theory of linkage or the experiment done by T.H. Morgan on Drosophila.